Maybe the network is better now. Now let me have any question? Any question? Any question? Can I continue now? Without question, can I continue? Can I continue?
Yes, sir. I'm not clear with that, the nucleotide. Sir,
Nancy,
I'm not clear with the nucleotide.
nucleotide. Okay. Any other question? Okay, now let me repeat what I said with regard to the nucleotide. Now you understood what is the nucleoside. Now in nucleotide is when you attach a phosphate group to a nucleoside, you have a nucleotide.
Okay.
Yes.
Phosphate.
Now the nucleotide can, the phosphate can be attached in two different places. It can be attached to carbon 3 of the ribose sugar or carbon 5 of the ribose sugar. When it is attached to carbon 3, for example, it will be nucleoside 3 monophosphate. If it attached to carbon 5, it's going to be nucleoside 5 monophosphate.
Okay.
Depending on the number of phosphate group that is attached. If it is 1, it will be monophosphate. If it is 2, it's going to be diphosphate. If it is 3, it's going to be triphosphate. Is that clear?
Okay. Yes, sir.
Now you have adenosine. Now adenosine, we know is a nucleoside containing adenine and a ribose sugar. When you attach the one phosphorus or phosphate group rather, which is called adenosine monophosphate. When 2 is attached, it's going to be adenosine diphosphate. When 3 is attached, it's going to be adenosine diphosphate. So usually in naming a nucleotide, this YLETE is attached. So instead of you saying adenosine monophosphate, you can say adenylate. If it is guanosine monophosphate, you can say guanylate. In the case of pyrimidine, if it is cyclic monophosphate, you can say cyclidylate. Instead of saying thymidine monophosphate, you can say thymidylate. Instead of saying uridine monophosphate, you can say uridylate. Is that clear?
No. Yes, sir.
Is that clear?
Yes, sir.
Good. So this is just the structure of the nucleotide. Remember, I said starting with the nitrogenous base, you can see the nitrogenous base here. You can see the nitrogenous base here, which is adenosine. When the adenosine attached to a deoxyribose, we have deoxyadenosine. So you have this nitrogenous base with the sugar, you have the adenosine. When one phosphate is attached through a phosphoester bond, we have deoxyadenosine monophosphate. When it is 2 phosphate is attached, we have deoxyadenosine diphosphate. When 3 phosphate is attached, we have deoxyadenosine triphosphate. Polynucleotide. Oh. In polynucleotide is when you have this nucleotide attached to one another to form a chain, a long chain of nucleotide. Did you understand?
Yes, sir.
So usually what happens, remember, while explaining the attachment of a phosphate I told you that the phosphate group can be attached to either a 3' or 5', right? So when you have a particular phosphate, a nucleotide, where on the 3', on the 5', the phosphate group is attached. You can see it here in red. You can see my cursor, right? Right?
Yes, sir.
Yeah. So you have this phosphate group attached to it. You have this phosphate group attached to it already at 5'. So in a polynucleotide, usually in DNA, like I said, usually they exist in monophosphate, not in triphosphate. So you only have one phosphate attached. So let's just assume this is only one phosphate, while the other phosphate remove them. Okay, are you there?
Yes, sir.
Okay, so you can see on the 5', there is a phosphate attached, but on the 3', there is nothing attached. So when you have many of these adenosine monophosphates, you have many of them. How do they come together, link to form a polymer? So you have this 3'. This 3' can also bind with another phosphate group from another nucleotide. For example, assuming, like I said, this is the only phosphate group that is attached. So this phosphate group can now go and attach to another nucleotide that contain this phosphorus, the ribose sugar, and the nitrogenous base through this 3'. So this 3' can react with another phosphate group of another adenosine monophosphate like this, until you have a very long linear chain called polynucleotide. Are we together?
Okay. Yes, sir.
Yes, sir.
No, sir. So I didn't get it, please.
In polynucleotide, remember, I said the phosphate group can be attached to two positions. Can be attached to 3' of the ribose sugar or the 5' of the ribose sugar. So usually it is attached to the 5' of the ribose sugar. So let's assume that nucleotide is adenosine monophosphate. So you have adenine, you have a ribose sugar, and you have the phosphate group attached to 5' carbon. Is that clear?
Yes, sir.
Yes.
Many of this adenosine mononucleotide. And this adenosine mononucleotide, they are going to form a polynucleotide. And we say in polynucleotide is the polymer of a nucleotide. So you have adenosine mononucleotide, another adenosine mononucleotide will link together to form a bond. You have two adenosine mononucleotide called dinucleotide. Another adenosine mononucleotide will now attach itself to this dinucleotide to form a 3 nucleotide, trinucleotide. Another one to form tetranucleotide. Another one pentanucleotide, like this, until it's formed a very long one we call a polynucleotide, right?
Okay, yes, sir.
Now, how is this binding happen? We said the first one, adenosine mononucleotide, have this 3'. So this 3' can now react with the phosphate group of the other nucleotide. So the phosphate group of the other nucleotide, adenosine monophosphate, that phosphate will now attach to this one. So remember that one also, it has this 3'. So the 3' of that, the second nucleotide, will also attach to another phosphate group of another new nucleotide. So that new nucleotide also have a 3', and that 3' can also react with the phosphate group of another nucleotide, like this, until you have a very long polymer of a nucleotide. Did you get it?
Yes.
Did you get it?
Yes, sir.
Okay. There is a situation you can have what we call a cyclic nucleotide. So usually I was explaining adenosine monophosphate. So when you have adenosine monophosphate, you have the phosphate group attached to carbon 5, right?
Yes.
Remember, this phosphate group contain how many oxygen? Contain 4 oxygen, right? 1 oxygen double bonded, while the 2 oxygen, they have a hydroxyl group attached to it. So it is one of them that is involved in this bonding with carbon 5. Another phosphate group of the same, another hydroxyl group of the same phosphate group can now in turn also react with this carbon 3 to form a kind of cyclic. It is attached to carbon 5 and at the same time attached to carbon 3. This is what we call a cyclic nucleotide. When it is adenosine monophosphate, it forms what we call a cyclic AMP. If it is guanosine monophosphate, it forms what we call cyclic GMP. Usually these molecules, they are a signal molecules. They are like hormones. They are like hormones. I think in physiology, you talked about endocrine hormones, different type of hormones, exocrine hormones. I think you talked about those things, signal molecules. So these are like hormones that are produced and they act where they are produced. So once they are produced, they act within the area they are produced. They are a signal molecules. So we talked about the function of nucleotide. One of the function of nucleotide, which we are trying to explain here, is that they form the building block of DNA and RNA. We all know that, right? Another one we explain is that they are carriers of chemical energy. For example, ATP. I already explained ATP and how ATP has been used as a major source of energy in the system, right? So ATP can also be GTP, UTP, CTP, but most commonly ATP is used. But either of them can be used as a source of energy. Another point, as a component of enzyme like NAD, FAD, for enzyme A, you will come to see what is NAD and NADP, FAD when you are doing metabolism and what role they play. Usually they are involved in oxidation reduction reaction. Or they serve as chemical messenger, as I explained the cyclic AMP, the cyclic. Now, do I have a question? Do I have a question? I have a question, class.
Yes, sir.
Go ahead.
It's about the functions of the nucleotide. I think the third functions, we should talk about cofactors. I don't understand that point.
In cofactors, shortly, I think in when we are discussing enzymes, there is an aspect where we'll discuss enzymes and their function, the enzymology aspect, which is, I think, toward the end of this class. We are going to talk about enzymes and their function. And we talked about there are some enzymes that contain some molecules we call cofactors. These molecules, they help the enzyme to carry out its own function. So some of these cofactors, there are molecules that help these enzymes to carry out their own function. So they are found attached to these enzymes. So some of them contain this nucleotide. They contain this nucleotide. And I gave example of FAD. FAD is flavin dinucleotide. NAD is nicotinamide adenine dinucleotide. Is that clear?
Yes, sir.
Is that clear, Daniel?
Yes, sir. Yes, sir.
Okay. Any other question? Hi, Patrick.
Sir?
Do you have a question?
No, sir.
Please. Listen. Kennedy, are you with us?
Yes.
Kennedy, are you with us?
Yes, sir. Yes, sir.
Aisha,
Yes, sir.
are you with us?
Yes, sir.
You have a question?
No, sir.
Mariam,
Yes, sir.
you have a question?
Yes, sir.
Okay. Salome. Salome,
Yes.
you have a question?
No, sir.
Okay. So please, I have recorded this part. I think the first part, which wasn't too clear, was we missed recording of that part actually. So I want to believe the previous recordings we have last year can serve for that. So I only have the recording of some aspect of the first study session we discussed today, and then the second study session. So I will share this as I did, and then for the other part, please you can look at any recorded video in the channel. Like I told you, I must have recorded last year class. So the recorded part of that will be available there and you can make up with it. In fact, I will look for it and try and share the link with you as I will do for these recorded ones. And like I said, at the end of all this, I have these questions, which is expected of you to go there and try and answer them as a quiz. And I will try and make some of these questions to be a compulsory or questions that will attract marks once you answer them. So I will try and do that. In the absence of any question, I will call it out for today. Thank you all for coming.
All right, thank you, sir.
Thank you,
We are
sir.
grateful.
Thank you, sir. Thank you, sir.
Thank you, sir. God bless you, sir.
Thank you, sir.
Thank you, sir. Who not chose the logo? Ah, very well. Yes, ma'am. Yes, ma'am. I'm so happy. Gbogbo yín ni mo ń rí bí mo ní àti gbẹ̀yìn náà.
you.